Polypeptide synthesis begins with transcription. It is where the DNA sequence of a gene is made into a molecule of mRNA. This all occurs in the nucleus of a eukaryotic cell. The RNA polymerase connects to a specific part of the DNA at the beginning of a gene. This tells the DNA to unwind, exposing the template strand for transcription. RNA polymerase reads the DNA strand and makes complementary RNA strands by adding nucleotides A, U, C, and G. Adenine pairs with uracil while cytosine pairs with guanine. The RNA strand is the same as a DNA coding strand, except thymine is replaced with uracil. When RNA polymerase reaches a termination sequence in the DNA, it stops transcription, releasing the newly formed pre-mRNA strand. The mature mRNA exits the nucleus and enters the cytoplasm where translation begins. Okay, so the bases move along the DNA strand like this, and then they go to the RNA transcript, and then they split off, and then this comes down to here, which gets it ready for translation. Translation begins with the mRNA getting DNA from the nucleus. It has a sequence of codons, each representing a specific amino acid. After the mRNA exits the nucleus, it arrives in the cytoplasm where it joins with the ribosomes. The ribosomes have a 5' to a 3' which tells the ribosome where to begin translation. The ribosome moves along the mRNA in the 5 to 3 direction, reading each codon. Each tRNA with the corresponding anticodon binds to its complementary codon on the mRNA in the ribosome. The ribosome moves one codon down to the mRNA, shifting the tRNA from the start to the exit site and releasing it from the ribosome while the next codon is getting ready to be translated. Translation continues until a stop codon, UAA, UAG, UGA, is reached on the mRNA. These codons do not code for any amino acid but instead signal the end of translation. When a stop codon is encountered, a release factor binds to the ribosome, causing the release of the newly synthesized polypeptide chain. Through translation, the sequence of nucleotides in mRNA is converted into a sequence of amino acids, which folds into a functional protein. This process is essential for the production of the wide array of proteins that carry out the myriad functions required for cellular life. These proteins ensure the process is precise, efficient, and well-regulated. This process, starting with transcription and ending with translation, explains how genetic information flows from DNA to RNA to protein. The bases move along the mRNA strand from the 5 to the 3, moving along, getting taken onto the tRNA, and then connecting with the proteins all the way down.